At present, probably only one student or viewer is connected. Today we are going to discuss on a very significant part for the competitive examinations, and kindly communicate. Only Sakshi is attached. Today is the significant part for the competitive examination, and we are discussing on the some important parts related with the macromolecule of protein, and today we observe or we discuss on the classification of proteins, configuration of proteins, and most important is the Ramachandran plots, and sometimes, particularly in the NET or in the GATE examinations, some questions are asked from the Ramachandran plots. We have already discussed on the amino acids, nature, classifications, nomenclatures of the amino acids in my previous classes, and in the section of the biochemical micromolecules, we all know that the protein is polymerized form of the amino acids. Of course, we all discussed or we all study in the under the heading of the biochemistry, and the term biochemistry was at first used by Newberg in 1903, and in my last sections when we discussed on the biochemical macromolecules, we have already acquainted or known that the macromolecules, molecular weight of macromolecules are commonly considered under the 10 to the power 3 to the 10 to the power 9 AMU or atomic mass units. That is the molecular weight of the biochemical macromolecules commonly. And just a minute. टुडे आज थोड़ा मेरा चश्मा जो है वो टूट गया है, थोड़ी सी कठिनाई हो रही है, आंखों पे लोड आ रहा है हमको. एनीवे, the macromolecule components commonly we are discussing on the macromolecule today. We are discussing on the macromolecules and mainly the part related with the proteins. But before coming on the protein, I want to provide some basic idea of the protoplasm, because the protoplasm is also a macromolecule. We studied the protoplasm under the macromolecules, and we all know that the protoplasm, all the macromolecules including the water and other combinations of the uh, biochemical molecules present in the cell is the protoplasm, and it is the crystallo-colloidal structure, and its granules varies from 10 to 10 power minus 1 to 10 power minus 4 mu. It is the basic granules of the protoplasm, and the term protoplasm was at first used by Purkinje. It is the basic idea. But what is important related with the protoplasm, it is referred as the physical basis of life by the Julian Huxley. Wilson presented the colloidal theory of the protoplasm. O. Hartwig presented the protoplasmic theory. All are the historical significance, and today we all know that if the life is persisted, then protoplasm is present. It is necessary that the protoplasm is present, because the protoplasm is related with the all activities of the living components. And here, the colloid, that is the protoplasm, we observed and we referred as the fourth matter. We all know that the matter gas is divided into the gas, liquid, solid, and that is the fourth state of the matter, and it is in the physical science it is referred as the colloid, and in biological science we we referred it as the protoplasm. That protoplasm represents all the properties of the all the matters, like it represents the some property of the liquid, some property of the solid, and some property of the gas. In addition with, there is some difference between the physical body, that is the colloid, and the biological body, that is the protoplasm, because the colloid has the non-living activities. It is only responsible for the non-living functions. But in the protoplasm, in addition with the properties of all these components of the matters, it has the capacity of the to regulate the all properties of the living system. So the first important character of the protoplasm, that it is a colloidal particles, and if it is the colloidal particles, it is the contains two heterogeneous components. Protoplasm contains, it is the characteristic of the protoplasm that it contains two heterogeneous components, that is the dispersed phase and dispersing phase. Dispersed phase is the macromolecular components commonly, or the dispersion phase is the medium. It is generally the liquefied form. That means the protoplasm contains the two phases, and it has the two components, dispersed phase and dispersion phase. Second characteristics of the protoplasm, that it remains in the two forms, that is the liquid form referred as the sol, and in the granular form it is referred as the gel, and that is the gel and sol stages are interconvertible. It is not rigid. Third characteristics of the protoplasm is the Tyndall effect. It is the most significant. Of course, the Tyndall effect is also represented by the colloid, and the Tyndall effect represents that when we provide the light, if we provide and if we passes the light through the protoplasm, it gives a opalescent nature, opalescent nature, and when the light is blocked to the medium, it represents sometime glow-like a structure. That is the Tyndall effect. When it passes through the, when light is passes through the protoplasm, it represents opalescent appearance. That is the Tyndall effect. Next characteristics of the protoplasm is that the particles represent Brownian movement. The particles of the protoplasm represents Brownian movement. And the fifth next characteristic of the protoplasm, that here all biological properties of a living organisms are occurred. This one is the living property and only observed in the protoplasm but not present in the colloid. That is the basic difference between the colloid and protoplasm. And here the protoplasm is responsible for the regulating of all the biochemical properties, and it is also present both in the plant and animals. In anywhere where the life exists, there must be availability or presence of the protoplasm. Sometimes the protoplasm is liquefied, and that protoplasmic liquefied part of the protoplasm, particularly when we observed in the eukaryotic cells, that in the eukaryotic cells the protoplasm is divided into the nucleoplasm and cytoplasm, and nucleoplasm present in the nucleus, and cytoplasm is present in the in between the nuclear membrane and plasma membrane. And here the nature of the protoplasmic nature of the nucleoplasm and protoplasm differs only on the presence of the some proteinous component, a specific proteinous component in the nucleoplasmic mass and in the cytoplasmic mass. But in the cytoplasmic mass, some liquefied forms is observed, and commonly another term is used. Only the term is used. It is not any significant deviation or variation. That is the cytosol. And in the cytosol, all cell organelles are present or floated in the cytosol. That is some basic idea of the protoplasm, and the protoplasm comes under the heading of the macromolecules. Why I provide some basic idea of the protoplasm. Now we come on the topic of the protein. And in my previous class, I stated that the proteins are nothing but the polymerized form of the amino acids. That means the polymerization of the amino acid means some specific bond is present in between the amino acid and amino acid, that is responsible for the attachment of the more and more amino acids and form the proteins or polypeptide chain. Of course, we are confined our discussion in this streaming related with the biochemical part only, not with the biosynthetic and other regulatory parts of the proteins, because protein is most significant biochemical macromolecules of the living system, because the protein has a number proteins are responsible for the different functions, like it is responsible for the structural component, they are responsible for the recognizing the cell adhesion, that is the cell recognition, that it is responsible for the communication, it is responsible for the catalytic activities like enzyme, it is responsible for the intracellular trafficking, that is the chaperones, they are the small proteinous components, a small polymerized form of the amino acid, that is the chaperones. That means there are a number of the functions related with the proteinous component. So the present streaming we discuss on only the proteins, and I am just saying that we discuss the protein configuration of protein, classification of protein, and most significant for the NET, that is the Ramachandran plot. Now, we studied earlier, or all are studied at the intermediate level or the graduation level, that the amino acids are combined by the formation of the polypeptide chain in between the alpha carboxylic group of the alpha carbon of first amino acid and amino group of the alpha carbon of the subsequent amino acids by the removal of one molecules of water. And that formed bond is referred as peptide bond. And that means the peptide bond is responsible for the formation of the proteins. Conventionally, it is not biochemically or it is not the chemical parameters, but conventionally we divide biochemical molecules into peptides, peptones, and polypeptides. We considered the 2 to 20 amino acids chain as peptides, 21 to 50 as polypeptides, and when it is more than the 50, then it is referred as proteins. These are the conventional classification. It is not the rigidly defined, but it is the conventionally referred as the peptides, peptones, polypeptides. Peptones are some smaller components are referred as the peptones. Then, if we study the proteins, the sum, if you consider the nutritive point of view, then some amino acids are referred as essential for the man, because in commonly the microbial population, yeast, other one, they are mostly responsible for the synthesis of the amino acids. But in man, 10 amino acids are not synthesized in the body, and that 10 amino acids are referred as essential amino acids. That is the nutritive essential amino acids. And among these 10, 2, that is the arginine and histidine. Arginine and histidine are referred as the semi-essential amino acids because they are synthesized at the some level, some extent they are synthesized in the body. The 10 amino acids, the 10 amino nutritive essential amino acids are arginine, histidine, leucine, isoleucine, lysine, methionine, threonine, valine, and cysteine. These are the essential amino acids. And sometimes, one thing important, sometimes questions are asked in the examinations and the number are provided. Suppose in a question it is asked that a polypeptide chain has the 500 amino acids, calculate the molecular weight of the protein. And if such type of question is asked in the examination, you remember one point that on average, commonly on average, the weight of the amino acid is considered as 138 mu, 138. And when the polypeptide bond is formed, then it is responsible for the 1 unit of water. That means 16 and to 18. 18 weight value of the weight is removed, and it means you subtract the 18 from the 138, then the remaining value is the 120. That means the total, if we consider that in a question 500 number is placed, that means the bond present in the polypeptide chain is 499. That means 499 value is deducted from the 499 into 18 value is deducted from the total value, that is the 500 into 138, and the result represents the total weight of the proteins has the 500 amino acids. And that means it is a very easy question. You remember only that on average the weight of the amino acid is 138, and one polypeptide, if the one polypeptide chain is formed, then it is responsible for the deduction of 18 value, and now you calculate. Then if we consider the classification of the proteins, the proteins are classified on the basis of solubility, shape, and chemical composition. At first, on the basis of the nature of the chemical composition, proteins are classified into two parts. That is simple protein and conjugated protein. The simple protein is formed only by the amino acid. Non-amino acid components are not present in the simple proteins. And in the conjugated proteins, some non-amino biochemical components are present in the conjugated proteins. This differentiation is based on the nature of the chemical composition. Then the simple proteins are divided into two parts on the basis of the solubility. That is the fibrous protein and globular protein. In the simple proteins, if you consider the percentage of elements in simple protein, then sometimes the question is asked in the group 2, or if the some students of the NEET are connected in the streaming, I don't know who are connected or who are not connected, then the top question is also asking sometimes in the NEET examinations. Then here the percentage element of elements in simple protein is 50% carbon. Carbon is present 50%, oxygen is present 23%, nitrogen is present 16%, hydrogen is present 7%, and sulfur is present 0 to 3%. Because only two amino acids, that is the methionine and cysteine, has the sulfur content element. It is the percentage value of the elements in the simple protein, not in the conjugated protein. Because in the conjugated protein, the percentage may be varied on the basis of the association of the non proteinous component. And here, the simple proteins are divided into the fibrous protein and globular protein. The fibrous protein is again divided into the different categories like collagen, elastin, myosin, osin, keratins, and the collagens. Here again, the enzymatic activities and nature which is related with the heat is different in the simple proteins. The collagen is present in the connective tissues. The elastin is present in the elastic fibers, that is the yellow fibers, also present in the yellow connective tissues. The keratin is present in the hoops, nails, like these hairs. All are the very simple. Mycin is present in the muscles. Osin is present in the bones. And these are the nature of the fibrous protein. Commonly, the fibrous proteins are related with the structural component. Commonly, they are the structural component. Fibrous proteins, commonly, if you observe the fibrous protein, they are generally associated with the formation of the some structures. They are the structural proteins. Second is the globular protein. And globular protein is a soluble, and they are associated with the metabolic activities also, structure also, communication also, immunological protection also. There are different works of the globular proteins. The globular proteins are again divided into the different categories, like albumins. It is very easy. Albumins, if you eat egg, in egg, albumin is present. White part, that is the albumin. Globulins, immunoglobulins, in an antibody, that is the globular proteins. Globulins. Then is histones. Histone is also globular protein. Histone is observed in the DNA, eukaryotic DNA. That is the protamines. Prolamines. Glutins. Glutins is more important because here a number of research is associated with the glutins. That is prolamines. Prolamines present in the maize vegeta, like this. Protamines is present in the fish sperms. These are the different parts of the globular proteins. And on the basis of the activities, because globular proteins represent variation in the functions. Some globular proteins are associated with the structure also. Some globular proteins are responsible for the different metabolic activities. Some amino acids are responsible for the immunological activities. There are a number of the globular proteins. The next part is the conjugated proteins. Just I am saying that in the conjugated proteins, the proteins are present, that is the chain of the amino acids are present, and in association with the primary structure, the primary polypeptide chains, some non-peptidic components are also associated. And here, in the conjugated proteins also, the function is variable. Here also the function is variable. It is not responsible for a single functions. And on the basis of the non-proteinous component, on the basis of the chemical nature of the non-proteinous component, the conjugated proteins are further divided into the different parts. That is the metalloproteins, like in hemoglobin, metal is present. But here, just I am saying, if in your question the hemoglobin is asked and both choices are present, metalloprotein and chromoprotein, then it is referred, it comes in the heading of chromoprotein, not in the metalloprotein. So, if you reply in your examination, it is necessary to remember all the facts related with the any component. And here, a very minor difference is present between the metalloprotein and chromoprotein. If a metal is associated with any proteinous component and that metal is responsible for the appearance of any color, then it comes in the chromoprotein, in the heading of the chromoprotein, not in the heading of the metalloprotein. But if the metal is not responsible for the appearance of any color, then it is included in the metalloprotein, not in the chromoprotein. It is the very minor difference between the metalloprotein and chromoprotein. Likely, another is the mucoprotein. In mucoprotein, the carbohydrate components are present in association with the proteinous component. And here also, if a question is asked in examination, and in some books, if you go through the different books, variation, you commonly observe the variations in the representation of this type of facts. If a question is asked in your examination and it is referred that, what is the chemical nature of the antibody or IgG? And if in a question is mucoprotein is present, you easily select the question answer, the mucoprotein, because in the antibodies, some carbohydrate components are present. Why so? The mucoprotein represented the proteinous component plus the carbohydrate component. But here again, the glycoprotein, and another differentiation is the mucoprotein and glycoprotein. And the basic difference is again here. If the concentration or the percentage of the saccharidic component is up to 4%, then it comes in the glycoprotein, not in the mucoprotein. And in this sense, if the IgG is asked, the percentage of the saccharidic components in the IgG is up to 4%. And here, then the reply is changed, then IgG is the glycoprotein, comes under the heading of the glycoprotein, not under the heading of the mucoprotein. So it is necessary to study with the full concept. If you will not develop the proper concept, you will miss your question in your examination. Third one is the nucleoprotein, flavoprotein, phosphoprotein. These are the basic. Flavoprotein means has FAD, जहाँ पे flavin content show, that is the flavoprotein. So these are the differentiation of the conjugated proteins on the basis of the association of the non-peptidic components with the polypeptides. So these are the common facts, but it is necessary to understand properly the classification of the proteins. Now, after the classification, we come on the next part, that is the configuration of the proteins. And the configurational, if we observe configurationally the proteins, because the protein configuration is most important component for its biological activities. And if the configuration of the protein is altered or changed by any physical or chemical factors, then the protein is neutralized and the protein loses its biological properties. So the logically, and if you observe the configuration, the proteins are divided into four groups. That is the primary configuration, secondary configuration, tertiary configuration, and quaternary configuration. The proteins represent four types of configuration. At first, we comes on the primary configuration. It is very easy. The protein is synthesized by the translation, by the mechanism of translation, and the amino acids are associated in sequence as the sequence of the nucleotidic sequence present on the DNA. So, in the primary configuration, proteins represent sequence of amino acids. That is the specific, primarily the synthesized polypeptide chain represent sequence of amino acids. That means in the primary protein only polypeptide chain is present. Polypeptide bond is present, sorry. And the primary structure of polypeptide chain is most significant because it is determined the secondary configuration and formation of the non-peptidic bond. If we change the nature of the sequence or any one or two amino acids of the primary configuration, it will change the nature of the secondary configuration, folding of the polypeptide, further folding of the polypeptide chain, and that means the function of the primary chain may be distorted. So, the primary configuration of the protein is most significant. Second part, that is the in the primary protein, the only polypeptide chain is present. That polypeptide chain we just discussed is formed between the carboxylic group of the alpha carbon of the former amino acids and amino group of the alpha carbon of the subsequent amino acids. And that polypeptide chain has the covalent bond, is the covalent nature. ये covalent bond है. And when we study the secondary configuration of the protein, that secondary configuration represents folding of the primary configuration. The folded structure due to formation of non-peptidic bond. And basically, that is the hydrogen bond between amino acids of the primary chain. Of course, in some secondary configuration, interpolypeptide bond is also observed, and the bond which is observed and the bond which is responsible for the formation of secondary configuration is the hydrogen bond. Is the hydrogen bond. And the secondary configuration regulates folding of the main. In this way, the formation of the hydrogen bond regulates the folding of the main chain due to hydrogen bond formed between CO group and NH group. Their own local folding usually taken as aspiring of the polypeptide chain, and it forms loops like structure. It forms loop like structure, and that looping or bulging of the polypeptide chain is referred as secondary element. That secondary element, if you observe, they are of two types. That is alpha helix and beta pleated. And then alpha helix and beta pleated configuration represents variation in the nature of folding. In alpha helix, the polypeptide chain aspires to form aspires, and the aspiring is formed by the hydrogen bond, and it is responsible for the twisting of the polypeptide chain right-handedly, as we observed in the collagen or keratin. Then in the beta pleated, we observe the ladder-like foldings. Ladder-like foldings. And that ladder-like folding is formed between the formed by the hydrogen bond between the polypeptide chains and within the polypeptide chain both. And it is commonly observed in silk fibroin, thread of the silk, that is the silk fibroin, silk, natural silk. Of course, nowadays synthetic silks are also available in the market. That is the silk fibroin, that is the protein components. And that structure is referred as beta pleated structure. That means the secondary configuration is either alpha helix or beta pleated. The third structure, the third forms is the tertiary configuration. In tertiary configuration, further folding is observed, and that folding is formed by the formation of some extra other bonds like electrostatic bond, disulfide bond, van der Waals forces, electrostatic interactions like this. And the further folding gives rise to two famous molecular shapes called, इसको क्या बोलेंगे, fibrillar and globular. And that tertiary configuration is most significant biologically. Like if you observe the myoglobin, it represents a saucer-like structure, and it is the tertiary configuration formed by the further foldings. Again, the stage is observed, and that is the quaternary configuration. And in quaternary structure, further folding is observed and acquire the four planar structure. So, on page, it is not possible. Of course, the configuration, 4-degree configuration, we can observe on the computer programs. If we have the particular computer programs, we observe the 4D structures. That means in the quaternary structure, sometimes the in quaternary structure is formed by the two units or more than two units, and that component is referred as subunits, as we observed in the hemoglobin. Hemoglobin represents quaternary structure, and that quaternary structure is formed by two subunits, and one subunit is formed by one alpha and one beta polypeptide chains. That means the configurationally, if we divide the proteins, there are four possibilities, and we observe the four types. That is the primary configuration, secondary configuration, tertiary configuration, and quaternary configuration, on the basis of, what is the basis? On the basis of the spatial configuration of the protein. On the basis of the spatial configuration of the proteins. Now, if we observe the spatial configuration of the protein, sometimes a segment of the polypeptide chain, not the complete polypeptide chain, form their own dimensional folds. Of course, the complete polypeptide chain is responsible for the formation of the primary configuration, secondary configuration, tertiary configuration, and quaternary configuration. But sometimes a part of the polypeptide chain forms its own dimensional configuration. And that particular dimensional configuration is responsible for the particular biological activities, biological metabolic regulators, or biological recognition activities, or biological transportation activities, or any specific biological activities. That particular locally acquired configuration is referred as domain. And that domain is very significant, because if the protein present in same configurational stage, but if it nature of the domain is changed by any molecular or chemical change, then it loses its biological property. So, the study of domain, a study of particular domain, is most important. And here, today, a fast growing branch related with the research, a branch is growing very fast, that we called proteomics. That proteomics is responsible for the analysis of all these structures and stages. Change in the amino acid sequence in the primary configuration, or change in the configuration of the domain, or change in the nature of protein. That means it provides a significant point for the study, not only for the evolutionary aspects, but it provides an excellent field for the study of the regulation of the metabolic activities, regulation of the recognition activities, regulation of the communication activities within cell or outside cell, all this. So, the part is significant, and we will discuss further in the classrooms on these topics. We are just continuing on through the live कि आप लोग थोड़ा पढ़ाई में लगे रहें, ऐसा नहीं कि हम छोड़ दे तो आप लोग सो जाएं. क्लासेस होंगी, ऑफलाइन क्लासेस तो होंगी ही. एंड नाउ वी कम the most significant topic because it is new for you all, that is the Ramachandran plot, and it is present in your CSIR-NET syllabus. That is the Ramachandran plot. Of course, in classes, जो बच्चे क्लास में आ रहे होंगे, जो स्टूडेंट क्लास में आ रहे होंगे, बच्चे क्या आप लोग का सब अब बड़े बड़े हो गए, अब क्या बच्चे हैं, जो भी आप लोग क्लास में आ रहे होंगे, हमने बार बार बोला है कि द स्पेशियल अरेंजमेंट ऑफ द मॉलिक्यूल्स आर मोस्ट सिग्निफिकेंट. केवल उसका नाम, उसका कॉन्फ़िगरेशन नहीं, द ओरिएंटेशन ऑफ द मॉलिक्यूल्स इन द स्पेस, द ओरिएंटेशन ऑफ द बायोकेमिकल मॉलिक्यूल्स इन द स्पेस इज मोस्ट इंपॉर्टेंट. And that orientation is responsible for the biological activities. And here, the configuration is not as, of course it is significant, but not as significant as the orientation of the molecules in the proteins or in the polypeptide chain. That is the significant. And at first, three workers, Ramachandran, Ramakrishnan, and Shashi Sekharan, presented the concept of a spatial arrangement and torsion of the molecular torsion of the polypeptide chain, and that molecular torsion is responsible for the spatial arrangement of the molecules of the polypeptide chain on the planar, biplanar mood. दो प्लेन. That plot is referred as Ramachandran plot. Of course, that Ramachandran plot is presented in 1963, and at that time the Ramachandran plot was at first presented on the basis of the theoretical knowledge, mathematical calculations, and statistical analysis. But after the presentation of the Ramachandran plot, the plot was studied by the number of workers, and it clears from the study that the Ramachandran plot is most significant component for the analysis of in different techniques like X-ray crystallography, like these some techniques. We consider here only on the Ramachandran plots. Just I am saying that the torsion means the angle, bond angle of the proteins. If we observe, that means the Ramachandran plot. What is the Ramachandran plot? It provides a simple two-dimensional graphical representation of all possible protein structures in terms of torsion angle. That is the Ramachandran plot. And torsion angle, what is torsion angle? Torsion angle represents the angle of bond with respect to the peptide bond. That is the torsion angle. इसको हम समझाएंगे, आप लोग समझ भी जाइएगा, आप परेशान नहीं हो, थोड़ा ध्यान से समझें, इसको समझ में आ जाएगी बात. Just I am saying that although the Ramachandran plot was based on the theoretical concepts, but now it is most significant. Just we are discussing that the polypeptide bond is formed between alpha carbon of first amino acid, carboxyl group of alpha carbon of first amino acid and amino group of alpha carbon of subsequent amino acids. This peptide bond is covalent bond. We observe that it is a single bond, single covalent bond. But actually, it represents partial double bond character. And the bond length of the peptide bond, the peptide bond length is 1.33 angstrom. That is less than the single bond and more than the double bond. If we observe, because here the peptide bond is present, is formed between the carbon and nitrogen. Carboxylic group of the first amino acid. The carbon of the carboxylic group of the first amino acid, that is the alpha carbon of the first amino acid, and the amino group, that is the nitrogen, of the amino group of the alpha carbon of second amino acid. That means the bond is formed between, the peptide bond is formed between the carbon and nitrogen. And if we observe the single bond, that is sigma bond, between the carbon and nitrogen, it is commonly 1.49 angstrom. If we observe the double bond length between the nitrogen and carbon, it is of 1.27 angstrom. But here, the bond is formed between the carbon and nitrogen. The peptide bond is formed between the carbon and nitrogen, and bond length is 1.33 angstrom. That is less than the single bond and more than the double bond. And it represents a hybrid nature, that is nature of the primary bond and nature of the secondary bond. And if we determine the percentage, then it represents 40% nature of the second double bond. 40% nature of the double bond. The formation of the polypeptide bond is thermodynamically inorganic process. It is thermodynamically inorganic process, and here the delta G value, that is the change of energy, free energy, is plus 21 kilojoule per mole. 21 kilojoule per mole. Now we comes again on the nature of the bond. As the peptide bond has the 40% double bond nature, it is fixed. It is non-rotating. And then due to here change, the oxygen associated with the carbon of peptide bond developed partially negative charge, and the nitrogen associated with the alpha carbon of subsequent amino acid develops partially positive charge. That means here we observe only the peptide bond, and we observe simply that the polypeptide bond is formed between the carbon and nitrogen, carbon of the first carbon and nitrogen of the carbon of the carboxylic acid of first amino acid, and nitrogen of the amino acid of amino group of the second amino acid is not so simple, because it includes now here the 6 atoms are included here. Which one? Alpha carbon of first amino acid, carbon of carboxylic group of first amino acid, oxygen associated with the carboxylic carbon of the first amino acid, nitrogen associated with the alpha carbon of second amino acid, hydrogen associated with the alpha carbon of nitrogen of the alpha carbon of second amino acids, and alpha carbon of second amino acids. That is the 6 elements are associated here. And the torsion angle is determined, and that torsion angle represents the number of biological properties of the proteins. So the Ramachandran plot is the most important. And here these 6 carbons, if we observe these 6 carbons, these 6 elements, I just simplify all these 6 components for the proper understanding into 4 parts. Which one? The oxygen associated with the alpha carboxylic group of the alpha carbon, carbon of the carboxylic group of the alpha carbon, nitrogen of the alpha carbon of second amino acids, and rest components. That means if we simplify these in the 4 components A, B, C and D. B represents carbon of the carboxylic group which participated in the formation of the polypeptide bond. C represents N of the carboxyl group of the second, N of the alpha carbon of the second amino acids, that is C. Here, rest components of the alpha carbon of the first amino acids is A, and all components of the second amino acids associated with the alpha carbon is D. That means we observe here that B represents the carbon associated with the polypeptide formation of the polypeptide bond. C represents nitrogen associated with the formation of the polypeptide chain. Here, rest components attached with the B, now we call B as the A, and all components associated with the C is the D. That means here all we observed as the A, B, C and D, where in between the B and C polypeptide bond is present, and it is non-rotatory due to the nature of the double bond. So there is chance of the rotation of the only 2 points, that is rotation of the A, rotation in the angle of the A associated with the B, or the rotation in the angle of the C associated with the D. There is 2 rotatory points, and that rotatory points is referred as torsional angle and represented by phi and psi. So the Ramachandran plot is also referred as phi and psi plot. Ramachandran plot is also referred as phi and psi plot. And here the analysis of the phi and psi plot is referred, determination of the torsional angle of the phi and psi is referred as Ramachandran plot. That is the main point. Here again, when we comes the nature of the component, the planar configuration are represented either in cis or in transform. As I think that you all are properly understand the nature of cis and transform. Cis means the chemical components are present in one plane. In transform, the components are present in opposite plane. Here, with this rotational angle, one is phi and another is psi, is present between carbon and oxygen. One angle is present between the carbon and oxygen in the carboxyl group. Another is present with the nitrogen and hydrogen. That is all components. And if we observe theoretical point, then the rotation about C and N is possible, when the cis and trans movement, that is the rotation of the torsional angle, is possible if you observe in the transform, that is in the opposite direction. That means it is of the 180 degree, because main axis is the peptide bond. Main जो plane is determined by the peptide bond because it is the non-rotatory. And here, if it is present in the cis form with respect to the other components, then the degree, rotational degree becomes 0 because present in the one plane. And if present in a transform, that is the rotational degree is 180, that is the opposite plane. And here the peptide bond is separated, and that is referred as omega, by the omega, Greek letter omega, W, that is omega. That the degree value of the omega, that is actual the bond of the bond formed between the C and A, N, is 0 to 180 degree. That is the cis to trans. And I am just saying, if we observe in the biological system, the trans form is more prominent than the cis form. Why? Just we are coming on the topic. Trans form is more formed, and if we observe the ratio of the trans and cis, then the ratio is 1000:1. But one point is here, which you all remember, that the value clearly represents that the orientation of the orientation with respect to the peptide bond is more preferred as the trans, because the value is ratio value we observed commonly in the living system is 1000:1. But in proline, the nature of the amino acid is changed, and if the proline is present in the polypeptide chain or involved in the formation of the polypeptide, then it prefers the cis configuration, not the trans configuration. And here, that is the omega peptide bond, and from the omega angle, the amino group 1, amino group represents the torsional variation, and that is the phi. And second is the carboxyl group torsional angle. Here the two torsional angle. Just I am saying that now the one torsional angle is formed with respect to amino group, and it is represented by the phi. Another torsional angle is formed with the carboxyl group, that is represented by psi. Then, which factor is responsible for the determination of torsional angles? Theoretically, phi and psi have any value because it is free and has capacity to orient in the space. Space is free for all molecules. Then theoretically, the phi and psi have any value between plus 180 to minus 180 degree. However, not all combinations are possible. Theoretically, it has possibility that the torsional angle may be present from plus 180 degree to minus 180 degree. But the combination is not possible. Why? Due to the physical classes of atoms. If the atom is present, then atom requires some space because it has electron clouds, nucleus, and above the nucleus it has the electron clouds. Then it requires some physical space. Then two atoms cannot occupy the same space. And due to prevent such type of physical clashes, they provide a limit for the torsional angle, and these torsional angle, these factors which prevent the physical classes create a force. That force or a factor, that factor is not force, force is not accurate, a factor, that factor is referred as steric interference. That factor is referred as steric interference. And that steric interference is responsible for the determination of torsional angle. Most values of phi and psi are therefore not allowed due to steric interference. And steric interference of non-bonding component, because components are involved in the bond formation, but non-bonding components provide the steric interference, and then steric interference is responsible for the determination of torsional angle, and that torsional angle determines the planar structure, two planar structure of the molecules, and that planar structures are represented on the y-axis, that is x and y, and that representation is referred as Ramachandran plot, and that Ramachandran plot may be represented or may be called phi and psi plot. In between, the phi is represented on x-axis and psi is represented on y-axis respectively. And they are mostly in transform. Then one thing further which you remember or keep in mind, in addition with the geometrical isomerism, just ion cis and transforms we are discussing here in the Ramachandran plots, the amino acids also represent the optical activities, that is the dextro and levo stages, D amino acids and L amino acids. Of course, in biological system, L amino acids are more prevalent and biologically active. And that L amino acids, the uh, molecular arrangement of the D amino acids and L amino acids are again different. That means the Ramachandran plot of the D amino acids and L amino acids are also different. The Ramachandran plot of D-alanine is not same as the L-alanine due to the steric interference. And steric interference is created by the spatial and molecular arrangement of the Ramachandran, of the molecules associated with the bonded elements. So, in this way, they provide a accurate spatial arrangement of the molecules, and they are represented by the Ramachandran plots. And as I think that the students or any viewers, if so, are understand the Ramachandran plots, and if anyone has any problem related with the Ramachandran plots, he or she is free to comment or make any query if you have. And you all are free to communicate whatsoever you feel. थैंक यू, आप लोग जुड़े इसके लिए बहुत बहुत धन्यवाद।